Hi guys, welcome to our channel of Sciences and Mathematics. My name is Ernest. Today we are going to look at one of the founding principles of the concept of genetics. And this basically will entail different concepts from the perspective of chemistry and biology. And I'm going to mention a few of which we have already discussed in some of our videos. But for this case particularly, I'm going to look at the foundational structure of the main hereditary bases and nucleic acid that we're going to look at. And that basically consists the concept of DNA and RNA. Now, in this case, we're going to look at how these basic nucleic acids can be defined insofar as chemistry is concerned. So in this case, I'm going to look at the concept of carbohydrates, which are basically going to form the basis or the foundation structure for this DNA and RNA. Since we're going to see that basically these two nucleic acids are derivatives of these carbohydrates. So in this case, I'd like to welcome us. Kindly feel free to check on our comments section or maybe in some of our discussions on the playlist that I'm going to put in the link in the description box so that you can see some of our videos that we've already discussed on the same. So without further ado, thank you and welcome. So as I've said, today we're going to look at one of the main concepts which is going to assist us basically in forming the foundation of our discussions under genetics, and that is basically three fundamental areas which I want us to discuss. So before we study the concept of genetics, we need to understand some of these areas, because it's from this that some of the concepts that we're going to discuss, maybe in a figurative way, are going to be clear for us. So from our definition, basically, genetics refers to the study of inheritance and variation. And we're going to see that the concept of inheritance and variation basically is based upon this, what we call hereditary characteristics, which are going to be discussed under the concept of these two nucleic acids that we refer to as the DNA and RNA, which I want us to stress and try to see how they can be able to be represented, or what are some of the differences between these two basic nucleic acids. So in this case, I'm going to use a series of illustrations to try and achieve this objective. And one of them that I'm going to use maybe is concerning the concept of carbohydrates that we had discussed earlier in our previous video. So in our discussion of carbohydrates, we said that carbohydrates can be loosely defined as compounds or components which make up of the energy-giving foods. And we said basically they are made up of what we call the plant, or made up of cellulose and other materials, which basically is the cell wall of our plants, and which sometimes we can refer to as wood. So in this case, this is just a broader definition of how we can be able to interpret the concept of what we call carbohydrates. But basically, from a chemistry perspective, we're going to see that these carbohydrates can be defined in another way, which is going to shed light on basically what they are made up of. So in this discussion of carbohydrates, we say that they are made up of two broader categories, or what we refer to as types. And we say that these two types are the polyhydroxy aldehydes, and the next we said it is the polyhydroxy ketones. And from this sense, we said that we can be able to have a different naming system for these basic two types of carbohydrates. And the first one, we said that the ones which are derived from the polyhydroxy aldehydes are known as aldoses, and the other ones which is derived from the polyhydroxy ketones are known as ketoses. So therefore, from these two broader sense that we have already illustrated for this naming of these specific two types of carbohydrates, you can look at this specific name and try to see that they end in a characteristic way. So in this case, if you try to look at the suffix, it is starting, the prefix is starting with aldo, basically trying to imply the concept of aldehydes, and keto, trying to imply the concept of ketones. So if I may try to expand this one in a sense, we said that the classification of carbohydrates can be classified as either monosaccharides, and basically we said that classification is based on this is specific term saccharum, which basically is a word that refers to the concept of sugar. I presume this is a Latin word referring to the concept of sugar. Just try to check on that. So in this case, for just confirmation, so in this case we can be able to define these different classes of carbohydrates as either monosaccharides, disaccharides, and then we said about the polysaccharides, and then lastly we talked about the oligosaccharides, So basically these three different classes of carbohydrates have different properties, but from monosaccharides all through to the disaccharides, polysaccharides is a build-up of a class of monosaccharides which are joined together and perhaps having different characteristics at these two specific levels. So in this case, we're going to see that under the monosaccharides, we see that we can be able to classify these monosaccharides depending on the number of carbon atoms that make up that specific structure of that sugar. So in this case, if we have a carbon atom that is having, sorry for that, 5 carbon atoms, then we can be able to classify that one as a pent, and then, sorry for that, add a characteristic ending, in this case using the os. So therefore, a 5-member carbon for this particular sugar will be referred to as pentose. Now, I've basically mentioned that specific class intentionally because it's from this class of pentoses that we're going to have to derive our basic nucleic acids, which are going to be used in this specific discussion, which are known as the riboses or the ribose sugars. So from this specific class of pentoses that I've referred to as 5-member carbon, therefore we are going to see that we have sugars which are known as riboses. So therefore a ribose is going to refer to a sugar of a pentose class. So therefore, from that specific classification that I've left there for pentose, so therefore a ribose is a sugar of a pentose class, which basically occur widely in nature and constitutes the nucleosides, and then what we call the vitamins and enzymes. And we're going to often come across another specific term that is referred to as the nucleotide, which we need to contrast with this specific first term that I've listed on the board. So in this case, if these pentose sugars have got this specific characteristic or maybe have a relationship with this range of specific compounds, there's a difference between the term nucleoside and nucleotide. If we can be able to look at this characteristic ending for side and tide. So therefore, to create a difference between the two, the first one, which is a nucleotide, basically is made up of what we call a nitrogenous base in its structure. We have a nitrogenous base, and then in addition to that, we have what we call a sugar, which is a range of the sugars we have discussed. And then we have what we call a phosphate molecule, or we have a phosphate in this specific structure for the nucleotide. Now for the nucleoside, we're going to see that it basically consists of the base, which is a nitrogenous base plus the sugar. So in this case, the difference between the two is that the nucleotide consists of a phosphate, which is lacking on the side of a nucleoside. So in any case we come across the two, I hope that the difference between that specific compound, this one and this one, is going to be clear for us. So if I may move ahead, we're going to see that these ribose sugars are going to be sometimes referred to as deoxyribose. So from the structure, I'm going to give the difference, a figurative or maybe an illustrative difference between this specific deoxyribose and the ribose sugar. Now from the drawing that I'm going to have, it's going to show the difference between the two insofar as their chemical structures are concerned. Now the structure I illustrated on the board basically are the cyclic forms of this specific sugar, so the amino acid forms of these sugars for a 5-member ring that we have referred to, a 5-member carbon that we have referred to as ribose. Now if we try to look at this specific structure for this and this one, we see that we can be able to represent the cyclic form of these specific sugars in fissure projection using this specific kind of arrangement. So in this case, if we try to look at the first set for this specific molecule and the second one, actually much of the structure of this specific molecule is the same, only that there's a difference at this specific point where this first molecule has got oxygen and the second molecule has got hydroxide, that substitution at that specific point for this carbon. So in this case, this gives us the difference between the two names that we are seeing here. Now the concept of the deox that is added to this name of the ribose implies that this has been derived from the ribose sugar by the loss of a hydroxide, that particular hydroxide group that is attached at this point. So in case if we mention the concept of a deoxyribo or the ribose sugar, the difference in their structure basically is that hydroxide group and the hydrogen that is contained in the deox respectively. So in this case, this is a loss of hydroxide that is based at this specific point of this carbon in these two structures. So basically in our discussion, we're going to use the term ribose or ribonucleic acid, which I wanted us to understand where this specific term comes from. Now next we're going to look at one of the areas also that we need to check out, which basically is a representation of the cyclic form of carbohydrates, since they are going to bear importance in trying to represent these specific nucleic acids that we have referred to as DNA and RNA. So basically these cyclic forms of carbohydrates comes in a specific way, and we're going to see that among these carbon molecules that we have illustrated on the board, we are going to see that there is a specific one that is going to be involved in the rearrangement, and basically we're going to refer to that one as a numeric carbon. Now I'm going to have a fissure projection of a molecule and maybe a condensed formula structure. We talked about the Lewis condensed and then the expanded structure for that case. Now I'm going to have a condensed formula and then the fissure projection of a molecule, and then we'll see how that specific molecule of is going to have a rearrangement in order to form a cyclic carbohydrate, which is basically going to give us a clue on how the DNA molecule has been arranged. Now I'm going to use that condensed formula for this specific structure of this specific molecule. Now I want to form a cyclic sugar by the name of furanose, and therefore I'm going to look at this specific structure and try to see how I can be able to achieve that. Now in this case, if you try to look at this specific molecule, it consists of two functional groups. The first one is ROH group. This is the alcohol, and then we have what we call this specific group that we refer to as an aldehyde. So therefore, this is specific point. So in this case, these are the two groups which are going to get involved in the rearrangement for us to attain an aminoacetal form of or a cyclic form of this specific compound. So we're going to have this specific rearrangement taking place. Just to draw those carbons to show those specific points. So we're going to have this OH group, this specific point, and this is the carbonyl carbon for the aldehyde that I'm going to show at this specific point. So we're going to have a rearrangement whereby we're going to have 1, 2, 3, 4 carbons, 1, 2, 3, 4. So that basically represents a rearranged formula for this specific compound, and therefore we're going to show the reaction mechanism which is going to result in the formation of a cyclic form, whereby we're going to have this oxygen containing those two lone pairs of electrons. They're going to have a nucleophilic attack at that specific point of that carbonyl carbon, and therefore that bond is going to move in that direction, and the pairs on this particular oxygen are going to pick on that proton, and this is going to restore stability at that specific point. Therefore, in this case, we're going to have a resulting molecule which is going to have the structure shown as the one below. So in this case, I've just tried to show the number of hydrogens which are in this specific part of the molecule, so that we can be able to look at how the rearrangement has just happened. So in this case, we're going to see that this specific kind of or maybe carbonyl carbon that was located at this specific point has undergone a rearrangement with this oxygen from the OH group in order to form a cyclic form, which is going to be referred to as the Pharynx. So therefore, we can be able to achieve a cyclic form of these specific sugars through a rearrangement that we have seen using this specific mechanism. And therefore, these are going to appear widely in our discussion on the concept of genetics, whereby we're going to use the ribose sugar that I mentioned about in form of a cyclic form. And at this specific point, this particular carbon that I've illustrated is known as the anomeric carbon that are basically mentioned in the beginning of a discussion of this specific concept. So we're going to see that the sugars, which are basically, we said that they are involved in the nucleotides, are going to be attached at a specific point in this nucleic acid that we have mentioned, or they're going to have a specific bonding, characteristic bonding with this nucleic acids in the structure of these nucleic acids that we're going to try and see. So in this case, I'm going to also have a summary of what we have discussed, so that we can wind up on this. So our next video is going to pertain the concept of the structure of the DNA and RNA, and we're going to see how the various sugars, the concept of the sugar, the base, and the phosphate molecules are going to be attached respectively. So subset, basically, we have from the concept of carbohydrates, what we call the class of carbohydrates known as monosaccharides. And the monosaccharides are named using the term saccharum, which basically refers to the concept of sugars. And from using the concept of carbohydrates, we see that we have two categories, which are the polyhydroxy aldehydes and polyhydroxy ketones, whereby we have aldoses and the ketoses. And from the concept of aldoses, we see that we have a special kind of a class of aldoses, which are known as pentoses, which are 5-member carbons. Now, from these pentoses, we have a sugar that we refer to as ribose, which is widely found in nature in form of maybe vitamins, in form of nucleotides, and perhaps the enzymes that we mentioned. And we said that from this ribose, we have what we call deoxyribose sugar, which basically, from the illustration that are given basically implies that there is the molecular formula for the ribose and the oxoribose is the same, only that there's a difference for the ribose. The ribose has got a hydroxide group, while the dioxoribose lacks a OH group. So that's the main difference. And from that one, we already said that from the concept of these specific sugars, we said that we can have a cyclic form of carbohydrates, which are derivatives of the Fischer projection of the sugars, which can undergo a rearrangement to form the cyclic forms, which basically have illustrated in our last discussion and said that we have a specific carbon that we refer to as a numeric carbon, which comes from this is the aldehydes, which undergo a rearrangement together with the OH group to form cyclic forms of these specific sugars. So in this case, we're going to mention maybe the concept of sugars, bases, and the nucleic acids respectively. But in our next discussion, I'm going to show the formula, or maybe perhaps the illustration for the bases in nucleic acids for the DNA and RNA respectively. So I hope that this is going to assist us in trying to understand the chemical aspect or the structural aspect of this DNA and RNA before we go further into the concept of genetics. So that is it for this discussion, guys. Bye. God bless you.